Good morning, ladies and gentlemen. Today I'll be speaking about maximizing proliferation of Lactobacillus acidophilus and Bifidobacterium animalis subspecies lactis BB12 in vitro by integrating different sources of prebiotics. This is the overview of the presentation: introduction, research objectives, methodology, results and discussion, conclusion, and suggestion for further study. Moving on to the introduction, as all of us know, the increasing need and trend for functional food in current society has caused a remarkable change in the food pattern, which have urged the use of different nutraceuticals and production of functional food ingredients. Meanwhile, understanding the importance of maintaining a healthy gut system also have urged the use of several prebiotics as well as probiotic supplements, which possess a huge market potential. In context of maintaining a healthy gut microflora, prebiotics and probiotics plays a major role, where prebiotics are basically foods for probiotics. In order to be classified as a prebiotic, a food ingredient should undergo certain criteria such as resisting digestion and absorption in the upper gastrointestinal tract, selectively stimulating the growth of beneficial bacteria, and rendering physiological health benefits for host. So probiotics are by definition live microorganisms. When administered in adequate quantities are capable of conferring better health for host, such as elimination of pathogens, stimulating the immune system, reducing the serum cholesterol level, vitamin B production, production of bacteriocins, and controlling diarrhea conditions. So the concept of this research is facilitating probiotics with prebiotics attribute to harvest the promised health benefits out of probiotics. So moving to the research objective, the objective of this research is integrating different sources of prebiotics such as fiber, polyphenol extracts, and non-digestible polysaccharide extracts in suitable quantities to maximize the growth proliferation of Lactobacillus acidophilus and Bifidobacterium animalis subspecies lactis BB12 in vitro, which are two major probiotics. Moving to the methodology, very first, the prebiotics have been extracted from the plants. Fibers used in this study was extracted from Musa sp. pseudostem. Polyphenolic extract was isolated from Sesbania grandiflora flower petals, and non-digestible polysaccharide extract was extracted from Artocarpus heterophyllus seeds. Then after, these extracted prebiotics was designed in a method to produce 11 treatments, where 8 treatments are integrated prebiotic treatments which contain all of the extracted prebiotics in different concentrations, and 3 treatments were non-integrated prebiotic treatments which consisted only one prebiotic ingredient at a time. So then the bacterial cultures which were used in this study was activated since they were present in lyophilized form. Bifidobacterium animalis subspecies lactis was activated in MRS broth containing 0.05% of L-cysteine hydrochloride, and Lactobacillus acidophilus was activated in MRS broth, where both were carried out under anaerobic conditions at 37 degrees Celsius. Then these 11 treatments were introduced to fresh MRS broth separately for the 2 test probiotics, and then it was inoculated with the bacterial species. The initial inoculum size of Lactobacillus acidophilus was 5.5 into 10 to the power 5 CFU per milliliters. The initial inoculum size of Bifidobacterium animalis was 6.3 into 10 to the power 4 CFU colony forming units per milliliters, and they were allowed to grow for 24 hours at 37 degrees under anaerobic conditions along with the prebiotic treatments, and they were subsequently plated on MRS agar to get the plate counts. Meanwhile, a positive control was maintained for both the test probiotics, omitting the prebiotic treatments. Moving to the results and discussion, the effect of these 11 prebiotic treatments on the test probiotics were assessed in terms of increased colony forming units per milliliter in liquid MRS cultures compared to the control. Minitab 16 software was used to analyze the data obtained and highlight the treatments which are statistically significant in causing maximum proliferation of the 2 test probiotics. So the counts of bacteria was given in log colony forming units as present in the table. So it's very clear that when you observe all the 11 treatments, they are significantly effective in causing growth compared to the control. And when you consider the integrated prebiotic treatments, which is treatment 1 to 8, treatment 2 is more effective on Lactobacillus acidophilus, and treatment 4 is more effective on Bifidobacterium animalis subspecies in causing maximum growth proliferation. And when you consider the non-integrated prebiotic treatments, which is from treatment 9 to 11, treatment 9 is more effective on Lactobacillus acidophilus, and treatment 10 is more effective on Bifidobacterium animalis in causing maximum growth proliferation. Also, these analyzed data clearly dictates that these integrated prebiotic treatments are more effective in causing growth proliferation on the test probiotics than the non-integrated prebiotic treatments. And also, these integrated prebiotic treatments which have caused the highest growth of the test probiotics can be employed in initiating favorable health benefits in host. So, as shown in figure 1, it is the first figure shows the lactobacillus plate counts which were obtained with treatment 2 compared to control, and figure 2 shows the bifidobacterium plate counts obtained when grown with treatment 4 compared with control. So, in conclusion, that what we can say is combination containing 2% fiber, 0.2 polyphenolic extract and 0.2 non-digestible polysaccharide extract, which is treatment 2, is capable of causing maximum proliferation of Lactobacillus acidophilus, and combination containing 2% fiber, 0.6 polyphenolic extracts and 0.2 non-digestible polysaccharide extracts, which is treatment 4, initiated highest growth of Bifidobacterium animalis subspecies lactis BB12. Therefore, it is clear treatment 2 and treatment 4 provide grounds for maximizing the proliferation of test probiotics, that where we can use this in permanent implantation of probiotics in gut. And when considered as separate entities in liquid culture media, it's clear that treatment 9, which only consisted Musa sp fiber, has greater potential to cause significant proliferation of Lactobacillus acidophilus. And in the case of Bifidobacterium animalis lactis BB12, treatment 10, which consisted only polyphenolic extract of Sesbania grandiflora flower, has the highest effect on causing maximum proliferation of Bifidobacterium. And I would like to suggest further study to be carried out to understand how these integrated prebiotic treatments will be affecting the growth of these probiotic test probiotics in vitro, and also the potential concentration of these integrated prebiotic treatments which can cause growth proliferation in vivo. Also, further studies are required to understand the biotransformation mechanism and biotransformated products of all prebiotic treatments when metabolized by these test probiotic stains. Thank you.
Thank you. Paper is open for discussion.
Yeah, it's banana.
Now, can you generalize to all plant and types now when you say Musa? So is there any specific species or?
No, generally it's you can get any of the Musa sp, because the literature says that the outer sheet of Musa sp, the fiber is they have been used this fiber for various probiotics in literature to test their. It's not specified this, but it's all the same fiber, because it's insoluble fibers that have been presented in the outer sheet of Musa. It's not specific.
So the others one, the Sesbania.
Yeah.
So what is the local name for Sesbania?
That is Khaturu Murungamal.
Okay, thank you.
How do you plan to do an in vivo study?
Sorry?
How would you plan to do an in vivo study?
In vivo study is actually planned to do by counting this fecal colony forming units, how it have been. That means if the bacteria are colonizing, definitely the increase, you can see the increment of bacterial colonies in the fecal matter as well. So that will be the initial stage, and then going for models how this works, and then so on.